Good morning, everyone. My topic for today's presentation is choline and psychiatry, and my moderator is Dr. Shelley ma'am. This is the sequence of the topics that we will be going through today. First, let's talk about the synthesis and metabolism of acetylcholine. Acetylcholine is formed in cholinergic neurons. So, we will talk about the synthesis and metabolism, the receptors, Alzheimer's disease, effect of neurotransmitters on acetylcholine, effect of acetylcholine on sleep-wake cycle, effect of NMDA on choline, and choline role in neuroplasticity. First, we will talk about the synthesis and metabolism of acetylcholine. Acetylcholine is formed in cholinergic neurons from two precursors, choline and acetyl coenzyme A. Choline is derived from dietary and intraneuronal sources, and acetyl coenzyme A is made from glucose in the mitochondria of the neuron. These two substrates interact with the synthetic enzyme choline acetyl transferase to produce the neurotransmitter acetylcholine. The rate-limiting step in acetylcholine synthesis is the high-affinity uptake of choline. Once synthesized, acetylcholine is taken up into vesicles by the vesicular cholinergic transporter. Once released from the presynaptic neuron into the synapse, acetylcholine is not recaptured by an uptake mechanism, as occurs with other biogenic amines. Instead, the termination of cholinergic transmission is primarily dependent on the breakdown of acetylcholine to choline and acetate, a reaction catalyzed by acetylcholine esterase, an enzyme that exists both in soluble and membrane-bound forms. As we can see in this diagram, acetyl coenzyme A is derived from glucose in the mitochondria, and choline is derived from dietary and intraneuronal sources. They interact with choline acetyl transferase to form acetylcholine. This acetylcholine is broken down into choline and acetate by acetylcholinesterase, which is present both intracellularly and extracellularly. Another cholinesterase is particularly present in glial cells and also terminates the action of acetylcholine. Now we are talking about the cholinergic pathways. Cell bodies of some cholinergic pathways arise from the brainstem and project to many brain regions, including the prefrontal cortex, basal forebrain, thalamus, hypothalamus, amygdala, and hippocampus. And other cholinergic pathways have their cell bodies in the basal forebrain, project to prefrontal cortex, amygdala, and hippocampus, and are thought to be particularly important for memory. This is a diagram showing the cholinergic projections from the brainstem, and they project to many different brain regions like basal forebrain, prefrontal cortex, thalamus, hypothalamus, amygdala, and hippocampus, while projections from the basal forebrain project to prefrontal cortex, amygdala, and hippocampus, and these projections are particularly important for memory. Now we will talk about the receptors of acetylcholine. They fall into two major classes, muscarinic receptors and nicotinic receptors, which are named for the respective naturally occurring agonists, muscarine and nicotine. Now, 5 types of muscarinic receptors, M1 to M5, have been cloned and all are present in brain. They are coupled to G proteins and either act directly on ion channels or are linked to a variety of second messenger systems. M1, M3, and M5 are GQ11 coupled, while M2 and M4 are GI coupled. Location of muscarinic receptors, M1, M3, and M4 are primarily located in the cerebral cortex and hippocampus and mediate some of the effects of acetylcholine on learning and memory. The M1 receptor is thought to be key to memory function in the hippocampus and neocortex, where it may facilitate dopamine release. The M4 receptor is thought to be involved in regulating the ventral tegmental dopamine neurons to inhibit dopamine release in the mesolimbic pathway and reduce psychosis. The M2 receptors are concentrated in the basal forebrain, where they function as autoreceptors to control synthesis and release of acetylcholine. The M5 receptor is expressed at low levels in the brain. M1, M3, M5 receptors are excitatory postsynaptic receptors, while M2 and M4 are inhibitory presynaptic autoreceptors, which prevent further release of acetylcholine. And M4 receptors are also thought to exist as inhibitory postsynaptic receptors. This is a diagram which is showing that M1, M3, M5 are excitatory postsynaptic receptors, while M2 and M4 are inhibitory presynaptic autoreceptors and prevent further release of acetylcholine. M4 receptors are also thought to exist as inhibitory postsynaptic receptors. Then we will talk about muscarinic agonists. One example is xanomeline, which is an M4-M1 agonist and decreases dopamine cell-firing in the ventral tegmental areas in preclinical studies and improves positive symptoms of psychosis in early clinical studies of schizophrenia. The same drug or others working by similar mechanisms could theoretically reduce psychotic and cognitive symptoms in Alzheimer's disease. Now we will talk about muscarinic antagonists. The prototypical muscarinic antagonists are atropine and scopolamine, which occur naturally in the plant Derry nightshade or Atropa belladonna. Ingestion of muscarinic antagonists leads to other effects resulting from inhibition of parasympathetic nervous system, such as dry mouth, constipation, and urinary retention, in addition to central effects such as cognitive impairment and in extreme cases, delirium. Muscarinic antagonists such as benztropine and trihexyphenidyl are employed to treat the motor symptoms of Parkinson's disease and are sometimes helpful in ameliorating tardive dystonia, which is a possible side effect induced by long-term use of typical antipsychotic drugs. Many psychotropic drugs possess unwanted muscarinic antagonistic properties, including some tricyclic antidepressants and antipsychotic drugs. Now we will talk about nicotinic receptors. These are ligand-gated ion channels located at the neuromuscular junction, autonomic ganglia, adrenal medulla, brain and spinal cord. Nicotinic receptors that are expressed in both neurons and muscle mediate rapid excitatory signaling, but differ in their subunit composition. And every nicotinic receptor consists of 5 subunit molecules arranged around a central channel that opens to admit ions when the receptor is activated. These various nicotinic acetylcholine receptor subunits are categorized into 3 general functional classes: skeletal muscle units, subunits, alpha 1, beta 1, delta, and epsilon, and standard neuronal subunits, alpha 2 to alpha 6 and beta 2 to beta 4, and subunits capable of forming homomeric receptors, alpha 7 to alpha 9, where all 5 subunits are the same. As we can see in this diagram, acetylcholine neurotransmission, it is regulated by ligand-gated excitatory ion channels, which are nicotinic acetylcholine receptors, and there are multiple subtypes of these receptors. And 2 of the most important ones are those that contain alpha 7 subunits and those that contain alpha 4 and beta 2 subunits. Nicotinic receptors have been implicated in several pathological conditions. For example, mutations in one of the nicotinic receptor subunit genes is responsible for a specific form of epilepsy, autosomal dominant frontal lobe epilepsy. And in addition, a nicotinic subunit gene mutation may be among the factors predisposing individuals to schizophrenia, and alpha 7 nicotinic receptors have emerged as a potential therapeutic target for the treatment of neurocognitive dysfunctions in schizophrenia. In Alzheimer's disease and Parkinson's disease, there is a significant loss of high affinity nicotinic receptor sites, which may contribute to the pathophysiological changes underlying the cognitive decline observed in these conditions. And the most obvious role of neuronal nicotinic acetylcholine receptors is mediation of reinforcing and addicting properties of nicotine in chronic tobacco and e-cigarette users, in addition to the symptoms of withdrawal experienced upon cessation of use. There are relatively few nicotinic agonists or antagonists that are used therapeutically. One such example is succinylcholine, a weak partial agonist at nicotinic receptors, which is used clinically during anesthesia as a paralytic agent. And galantamine is an allosteric potentiator of several subtypes of neuronal nicotinic receptor and a weak competitive reversible inhibitor of acetylcholinesterase, which is used in the treatment of Alzheimer's disease. Now we will talk about varenicline. Varenicline is a selective alpha-4 beta-2 nicotinic acetylcholine receptor partial agonist, which is now widely marketed as medication to treat nicotine dependence and as an aid in cessation of smoking. Nicotinic full agonists include acetylcholine, which is very short acting, and nicotine, which is very long acting, and they open the channel fully and frequently. By contrast, nicotinic antagonists stabilize the channel in the closed state but do not desensitize these receptors. Nicotinic partial agonists stabilize nicotine receptors in an intermediate stage that is not desensitized and where the channel opens less frequently than with a full agonist, but more frequently than with an antagonist. Thus, it can reduce the dopaminergic reward that would occur if a patient did smoke by competing with nicotine, but also reduce withdrawal symptoms by stimulating at least some neurotransmission. Now we will talk about Alzheimer's disease. Alzheimer's disease is the most common cause of dementia, and there are three pathological hallmarks of Alzheimer's disease seen in the brain at autopsy. First is amyloid beta, which is aggregated into plaques, and neurofibrillary tangles composed of hyperphosphorylated tau protein and substantial neuronal cell loss. In this diagram, we can see two of the major pathological hallmarks, which are plaques and neurofibrillary tangles. And the third major hallmark of Alzheimer's disease, that is neuronal cell death. Tau is a microtubule associated binding protein, and in its non-pathological form, binds to and stabilizes microtubules within axonal projections. Synaptic vesicles carrying neurotransmitters are normally transported along these microtubules to the synapse. And when hyperphosphorylated, tau is no longer able to bind microtubules. So microtubules become destabilized and synaptic dysfunction occurs. Hyperphosphorylated tau also forms paired helical filaments, which aggregate into neurofibrillary tangles, one of the hallmarks of Alzheimer's disease. As we can see in this diagram, normally tau in its non-pathological form will bind to and stabilize microtubules within axonal projections. And it is along these microtubules that synaptic vesicles, which contain neurotransmitters, will be transported to the synapse. But when tau is hyperphosphorylated, it will not be able to bind these microtubules, and this will lead to synaptic dysfunction. It will also form paired helical filaments, which will aggregate and form neurofibrillary tangles. Stages of Alzheimer's disease. In the first stage, which is pre-symptomatic stage 1, A beta begins to appear silently in the brain. This A beta is detectable in the pre-symptomatic stage using PET scans and radioactive neuroimaging tracers that label A beta plaques. And cerebrospinal fluid levels of A beta are also low at this stage of the illness because it is being deposited in the brain instead of leaving the brain. In the second stage, which is called as pre-dementia Alzheimer's disease or mild cognitive impairment due to Alzheimer's disease, or even prodromal Alzheimer's disease, neurodegeneration can be demonstrated by the presence of elevated tau protein levels in CSF, by atrophy on MRI, or by the presence of neurofilament light in CSF or possibly plasma. As neurodegeneration or neuronal loss progresses, tau levels rise in CSF. The third stage and final stage of Alzheimer's disease is dementia. To diagnose probable Alzheimer's disease by clinical criteria, the patient must first meet the diagnostic criteria for all-cause dementia. And in addition to that, the patient must have a dementia which is insidious in onset with clearly demonstrated worsening of cognition over time, and either an amnestic problem, which is problem with learning and recall, or a non-amnestic presentation, which is language, visuospatial, or executive dysfunction. This is the criteria for all-cause dementia. The first point is that patients should have a cognitive or neuropsychiatric symptom that interferes with ability to perform usual activities. Then there should be a decline from previous levels of functioning. It should not be attributable to delirium or a major psychiatric disorder. It should be a cognitive impairment diagnosed through neuropsychological testing or patient performance. And the cognitive impairment should involve two of the following: impaired ability to acquire or retain new information, reasoning impairment, visual spatial impairment, and changes in personality or behavior. This is a chart which shows the 3 stages of Alzheimer's disease. In stage 1, the cognition is intact despite elevated levels of A beta in the brain, as evidenced by both positive A beta positron emission tomography, and there is reduced level of A beta in the CSF. In the second stage, the clinical signs will appear in the form of episodic memory deficits, and and in stage 3, dementia will occur, and currently treatment of Alzheimer's disease symptoms does not typically begin until stage 3 and long after the actual disease onset. In the symptomatic treatment of Alzheimer's disease, the first drug is donepezil, which is a reversible, long-acting, selective inhibitor of acetylcholinesterase without inhibition of butyrylcholinesterase. Donepezil inhibits acetylcholinesterase in pre- and postsynaptic cholinergic neurons and in other areas of central nervous system outside of cholinergic neurons where this enzyme is widespread. A central nervous system action boosts the availability of acetylcholine at the remaining sites normally innervated by cholinergic neurons, but which are now suffering from a deficiency of acetylcholine as presynaptic cholinergic neurons die off. Donepezil also inhibits acetylcholinesterase in the periphery, where its actions in the gastrointestinal tract can produce GI side effects. Donepezil is easy to dose, has mostly GI side effects, and these are mostly transient. As we can see in this diagram, these are the CNS actions of donepezil, which it inhibits acetylcholinesterase, hence increases acetylcholine and reduces the symptoms of Alzheimer's disease. Rivastigmine is a pseudo-irreversible, intermediate-acting, not only selective for acetylcholinesterase over butyrylcholinesterase, but perhaps for acetylcholinesterase in the cortex and hippocampus over acetylcholinesterase in other areas of the brain. Rivastigmine also inhibits butyrylcholinesterase within the glia, which may contribute somewhat to the enhancement of acetylcholine levels within the central nervous system. Inhibition of butyrylcholinesterase within glia may be even more important in patients with Alzheimer's disease as they develop gliosis when cortical neurons die, because these glia contain butyrylcholinesterase, and inhibition of this increased enzyme activity may have a favorable action on increasing the availability of acetylcholine to remaining cholinergic receptors via this second mechanism. Rivastigmine appears to have comparable safety and efficacy to donepezil, although it may have more gastrointestinal side effects when given orally, perhaps due to its pharmacokinetic profile, and perhaps due to inhibition of both acetylcholinesterase and butyrylcholinesterase in the periphery. So this diagram shows the action of rivastigmine. It inhibits acetylcholinesterase, and it also has some selective action over inhibition of butyrylcholinesterase. Next is galantamine. It has a dual mechanism of action, matching acetylcholinesterase inhibition with positive allosteric modulation of nicotinic cholinergic receptors. Theoretically, the inhibition of acetylcholinesterase could be enhanced when joined by the second action of galantamine at nicotinic receptors, thus raising acetylcholine levels at nicotinic cholinergic receptors by acetylcholinesterase inhibition could be boosted by the positive allosteric modulating actions of galantamine. However, it has not been proven that this theoretically advantageous second action translates into clinical advantages. Now, this diagram shows the memory network and cholinergic projections. Normally, cholinergic projections from the hippocampus and neocortex reach the basal forebrain. When Alzheimer's disease begins to occur, there are plaques and tangles which will cause loss of these cholinergic projections and will result in mild cognitive impairment and early Alzheimer's disease. This cholinergic treatment will boost the memory network in early Alzheimer's disease as the cholinergic projections in the hippocampus and neocortex are intact. But progression of this Alzheimer's disease will later destroy the complete memory network in hippocampus, neocortex, and basal forebrain, and later the cholinergic treatment will prove to be ineffective. We can also target glutamate for treatment in Alzheimer's disease. Glutamate has been hypothesized to be released in excess once Alzheimer's disease develops, perhaps in part triggered by neurotoxic A-beta plaques and neurofibrillary tangles that release glutamate from normal inhibition by GABA, as GABA interneurons also degenerate. In resting state, glutamate is normally acquired, and NMDA receptor is physiologically blocked by magnesium ions. And when there is a normal excitatory neurotransmission, a flurry of glutamate is released, and the postsynaptic NMDA receptor is a coincidence detector. The postsynaptic NMDA receptor is a coincidence detector and allows inflow of ions if three things happen at the same time: neuronal depolarization, glutamate occupying its binding site on the NMDA receptor, and co-transmitter glycine occupying its site on the NMDA receptor. And if plaques and tangles will cause a steady leak of glutamate, this would theoretically interfere with the fine-tuning of glutamate neurotransmission and possibly interfere with memory and learning, but not necessarily be damaging to neurons. And hypothetically, as Alzheimer's will progress, this glutamate release could be increased to a level that is tonically bombarding the postsynaptic receptor and eventually killing of dendrites and then killing of full neurons due to excitotoxic cell death. Memantine is a non-competitive, low-affinity NMDA receptor antagonist that will bind to the magnesium site when the channel is open. Hence, it will block the downstream effects of excessive tonic glutamate release by plugging the NMDA ion channel, and this may improve memory and prevent neuronal death due to glutamate excitotoxicity. This blockage of NMD receptors by memantine can be seen as a kind of artificial magnesium, more effective than physiological blockage by magnesium, which is overwhelmed by excitotoxic glutamate release, but less effective than ketamine, so that glutamate system is not entirely shut down. Since its mechanism of action in Alzheimer's is so different from cholinesterase inhibition, memantine is usually given concomitantly with a cholinesterase inhibitor.
Actually, to wait in the
So since its mechanism of action is Alzheimer's is so different from cholinesterase inhibition, memantine is usually given concomitantly with a cholinesterase inhibitor to exploit the potential of both of these approaches and get additive results in patients. Now, as we can see, this diagram is showing the Alzheimer's excitotoxicity. And because this diagram shows that neurodegeneration, which is caused by plaques and tangles, it is causing a steady leak of glutamate, which will result in excessive calcium influx in postsynaptic neurons, which in short term may cause memory problems, and in long term, accumulation of these radicals may cause destruction of neurons. And in this diagram, we can see that memantine will block this downstream effect of excessive glutamate release, and thus can prevent these memory problems and free radical destruction of neurons. Next, we will talk about effect of dopamine on acetylcholine. Dopamine neurons in nigrostriatal motor pathway make postsynaptic connections on cholinergic interneurons. Dopamine acting at D2 receptors will normally inhibit acetylcholine release from these cholinergic neurons. Hence, when D2 blockers are given, that is when D2 blockers are blocked acutely in nigrostriatal pathway, the same pathway which degenerates in Parkinson's disease, dopamine can no longer suppress acetylcholine release, thus disinhibiting acetylcholine release from cholinergic neurons. And this excess cholinergic activity will lead to symptoms of drug-induced Parkinsonism, that is akinesia, bradykinesia, rigidity, and tremors. So as we can see that normally at the D2 receptor, the dopamine will inhibit acetylcholine release. But when we give D2 blockers, dopamine is not able to inhibit acetylcholine release, which will cause enhanced release of acetylcholine and may lead to symptoms of drug-induced Parkinson's. Next, we talk about effect of serotonin on acetylcholine. Normally, serotonin will cause inhibition of norepinephrine and acetylcholine release. Hence, when a patient is given 5-HT3 antagonists, that will increase norepinephrine and acetylcholine release. So normally serotonin will cause, so when serotonin is released, it will bind to serotonin receptors on GABAergic neurons, and this will release GABA onto noradrenergic and cholinergic neurons. Thus, normally it will cause, it will reduce release of norepinephrine and acetylcholine. When 5-HT3 antagonists are given, it will remove this GABA inhibition, and hence release of noradrenaline and acetylcholine will increase. Then we talk about effect of acetylcholine on sleep-wake cycle. Acetylcholine, normally when it is released from the basal forebrain into cortical areas, it will promote wakefulness in the patient. And orexin, which is a hormone which regulates the sleep-wake cycle, it stabilizes wakefulness through its regulation on acetylcholine. When we talk about effect of NMDA on choline, NMDA receptor-induced excitotoxicity has been hypothesized to mediate abnormal choline metabolism that is involved in alterations in membrane permeability and cell death in certain neurodegenerative disorders. It has been proposed that NMDA receptor activation will initiate a calcium-dependent and region-specific release of choline from membrane phospholipids that, if sustained, may lead to alterations in membrane permeability and subsequent cell death. This relationship between NMDA-evoked choline release and delayed excitotoxic cell death might explain the special vulnerability of cholinergic neurons that die in certain neurodegenerative disorders, because cholinergic neurons are unique in using choline from membrane phospholipids for acetylcholine synthesis, and additional reduction of choline, which is produced by NMDA receptor activation, may render them more vulnerable to cell death compared with non-cholinergic neurons. When we talk about choline role in neuroplasticity, acetylcholine in the brain will alter neuronal excitability, influences synaptic transmission, induces synaptic plasticity, and coordinates firing of group of neurons. An important role for both nicotinic acetylcholine receptors and muscarinic acetylcholine receptors has been defined in hippocampal synaptic plasticity, and these effects are mediated through intracellular signaling pathways of muscarinic acetylcholine receptors and nicotinic acetylcholine receptors. The ability of acetylcholine to influence synaptic plasticity and dynamics of local circuits can also occur through astrocytic control of synaptic calcium concentration following nicotinic acetylcholine receptor stimulation. These are my references.
Can you go back to the slide? You said that acetylcholine anticholinergic can be used in treatment of dystonia. Where did you, what is the difference for that?
False.
False. Yes. Clinical uses that anticholinergics are not starting. This 5:3 and this double, which increases the cycle.
Yes, sir.
Relevant.
Sir, I was not able to find the clinical relevance of this. Yes, sir.
This one.
Sir.
5-HT3 antagonism enhances norepinephrine and acetylcholine. Do you know any 5-HT3 antagonist? Do you know any 5-HT3 antagonist? How is it effective? Where do you find this? Serotonin antagonism thing. Antidote. That's the 5-HT3. I mean, all the awesome serotonin antagonism. And serotonin, you would think that immediately serotonin is antidepressant. So why, how is serotonin antagonism be actually effective? Choline acts in both peripheral centers. Lot of results psychiatry and choline. So it goes to the CNS. Very well. Representative good for your initial presentation. It is a very good presentation. I think that what you can do that your slides are good. You explained also in a good way. Some of the things you could have edited out. You could have made it easier. Okay. For example, NMDA, so these were a little bit out of your main most topic. So you can give a passing reference in that, but you don't need to describe. Although it was well done because you did cover dementia completely with the diagnostic criteria. So that part also we could have, you could have give a passing, like just not describe that. That was good. I can tell you that you described all the initial that these are the wordings that I'm talking. So when you are going from say in a presentation, when you're transitioning from one phase to another, so make a section header, just like a title header. So the listener know that okay, now you're moving on to the second topic, second section. Okay, now I will cover choline in memory, choline in dementia, choline in sleep, like that. So that you can. You said acetylcholine in sleep halt. Can you please go to that slide? Acetylcholine in sleep halt.
Sir, acetylcholine normally promotes wakefulness.
Promotes wakefulness. Okay.
Yes, sir.
That is why when you say anticholinergics, okay, we should not give them late in the evening. Okay, you don't want anyway. You don't want you can say EPS. Cholinesterase, anticholinergics. Anticholinergics, ideally they also promote wakefulness. Cholinergics are promoting wakefulness. Cholinergic mechanisms are promoting wakefulness. Also. How do they act on EPS? तो तुमने बताइए. Good presentation. We covered a lot of things, but clinical formulation. बस ये था कि say 5-HT3 SDS में सब लोग उसी में होते हैं फिर तुम्हें जो एंटागोनिज्म होता है अपन बोलते हैं कि कम करता है और कोऑपरेशन को बढ़ाता है एंटी साइकोटिक ठीक। प्लानिंग बॉडी ये आजकल कौन देखता है ऑनलाइन वगैरह का